today what we're going to do is that we will look at other irreversible enzymes in glycolysis. So which one are those? Huh? PFK1, then pyruvate kinase. So those are the other irreversible enzymes. Here is our F6P, and we're going F16BP. And which enzyme does it? So this is our PFK1, right? All right. And then if we go this way, which is, what enzyme will that be? Phosphatase. So specifically FBPase1, right? That's the phosphatase, fructose 1,6-bisphosphatase 1. So it will make F6P. And what do we need here to go ATP? We need ATP, and we're going to make ADP. And what about here? Do we need something? What type of reaction is this? Dephosphorylation. And can you simplify a little more? You're releasing a PI, right? So this is a hydrolysis reaction, right? So we need water. And so if you have F16BP, then what process it will go on to? Glycolysis. That's the first committed step of glycolysis. So if we have it, the only path for it is to go to glycolysis. And what about this? Gluconeo. Okay, gluconeogenesis. So now we need ATP here to activate the enzyme. But if you have too much ATP, then would you rather do this or do something else? Something else. So this will be inhibited by high ATP. ATP is needed to activate it. But if you have high ATP, then this process is actually going to be inhibited. Okay? So ATP is going to inhibit PFK1, high ATP, because if you have high ATP, then that is the signal is that there is the ATP need has been made. Then don't use glucose to make pyruvate, instead store it as glycogen. What about ADP? And then we'll look at AMP. And we'll look at citrate. And we'll look at F26BP. So these are the regulators of these processes. So what about ADP? If you have high ADP, then what is the signal? Activate. Why? Because the ATP level has dropped, right? What about AMP? Last class we talked about what? If you have high AMP, then we need more ATP. We need to generate ATP, and we can do that by doing glycolysis, right? So AMP is going to activate. And AMP will deactivate the phosphatase. If you have high AMP, gluconeogenesis is expensive, right? So we're running low on ATP, then we can't do gluconeogenesis because there's not enough ATP. So remember, gluconeogenesis has steps where ATP are needed. It needs energy, but we never run out of ATP. Like, it's all relative, high versus low. That's why it is, right? So high ATP versus low ATP. And we saw last class that the small change in ATP has a drastic change in AMP. So AMP is more of a allosteric regulator for these processes. Okay, citrate. What about citrate? Have we seen citrate yet? Not yet. So it is going to be a part of citric acid cycle, CAC. And so you're making citrate, when acetyl-CoA enters the CAC, you make citrate. And when you have high citrate, what is the indication? That we're going to make a lot of ATP, right? So then, would you rather make PFK1 active or inactive? Inactive, because high citrate, meaning already we are going to make a lot of ATP. So then don't utilize glucose to make more ATP, rather store it. So citrate, high citrate is going to deactivate PFK1. Then F26BP, this we have not seen yet, which we are going to see in a minute. So what is F16BP? That we already know, right? F26BP is, all you have to do is to, instead of 1,6, it is 2,6. So the second and 6 carbons are phosphorylated instead of 1 and 6. F26BP is not a glycolytic intermediate. It is produced only to regulate glycolysis versus gluconeogenesis. So we're going to look at PFK1 activity. So your y-axis is PFK1 activity. So I'm trying to plot Michaelis-Menten plot, right? So you need velocity, and then what's the substrate? X-axis is substrate. What is the substrate? Okay, so if you have low ATP, then the curve would look something like that. And if you have high ATP, then it will look something like that. So that is that Km, and then the other Km will be about here, right? And so we looked at here ATP, but now we will look at how F26BP changes the activity of PFK1 versus FBPS1. So what happens is that we already established that F26BP is an allosteric activator of PFK1. So if you have F26BP, then the Km for PFK1 will be lower. So it will look something like that. And if you don't have it, then it will look something like this. And so again, if you just look at the Km values, right? So here, if you have F26BP, then the Km is going to be much lower than its absence. So F26BP will upregulate the activity of PFK1. If you don't have it, then you need a lot of F6P to make PFK1 active. If we look at FBPase1 activity, which is the phosphatase, right? And what's the substrate for this? Okay, so that's the substrate, right? FBPase1 will be inactivated if you have high F26BP. So its curves will look something like that. So the first chart we looked at PFK1 activity, and the substrate is F6P. If you have F26BP, then PFK1 activity will be upregulated. So it will reach its Vmax, or rather Km, at a lower substrate concentration than the absence of F26BP. See that if you don't have F26BP, then you need a lot more F6P to reach the Km, right? On the other hand, if you look at FBPase1 activity, then if you don't have F26BP, then it is active. If you do have F26BP, then it is it takes a lot more substrate to make it active. So this is another way to regulate the glycolysis versus gluconeogenesis pathway, which is by specifically making F26BP. F26BP is not a glycolytic intermediate, right? It is not a substrate for any of the enzymes in glycolysis, right? F26BP is not an intermediate, but it is made specifically to regulate the activity of PFK1, ABPS1, so that glycolysis versus gluconeogenesis can be regulated.
Take F16BP and use like a mutase to make it?
Yeah, so no, we don't. So that's the next thing I'll talk about, how we make it. So what we do is we do F6P and then go to F26BP. So PFK2, the substrate is F6P, but it makes F26BP. And these are the two other enzymes we're also going to look at in a minute. And so what will be the reverse reaction? What enzyme will that be? ABPS2. So now we are only looking at F26BP, which is not glycolytic intermediate. We are only looking how its production versus hydrolysis is coordinated. So remember, if you have this, then you upregulate glycolysis because you upregulate PFK1 activity. And if we activate ABPS2, then you will upregulate gluconeo. So we are talking about 4 different enzymes. One is PFK1, ABPS1, PFK2, ABPS2. PFK1, ABPS1, they are part of glycolysis gluconeogenesis. PFK2, ABPS2, they are not part of glycolysis or gluconeogenesis, but they are really critical to either make F26BP or break it, and that way they can regulate glycolysis versus gluconeogenesis. So keep that in mind. This is a very distinct thing, F26BP and F16BP. Even though only change in the position of the phosphate, the way they regulate things are very different. Now we will look at specifically how the PFK2 and ABPS2 activity is regulated. So PFK2, ABPS2 is a bifunctional enzyme. So there is a single enzyme, but it has two parts. One part has the PFK2 activity, which is to make F26BP, and other part has the phosphatase activity to hydrolyze that F26BP to F6P. What I have drawn here is that PFK2, ABPS2, and if you see the lights come on, meaning that's the active part. So now PFK2 is active, and on the other hand, if you have this ABPS2 lights on, meaning that ABPS2 is active. If PFK2 is active, then which process will be upregulated, glycolysis or gluconeogenesis? If you have PFK2 active, then what are you making? F26BP. If you have F26BP, then which part is active? Glycolysis. And you're actually down-regulating gluconeo. On the other hand, if you have ABPS2 active, then what are you doing? ABPS2 active meaning you're hydrolyzing F26BP, right? So if you don't have F26BP, then which pathway will be active? Glycolysis or gluconeo? Glycolysis will be down-regulated, gluconeogenesis will be upregulated. And this switching of activity is triggered by phosphorylation, dephosphorylation. Remember last class we talked about fed versus starved state, right? So can you bring that into context here, bring into play? Why don't you guys talk for 30 seconds and give me some feedback? last class we discussed phosphorylation, dephosphorylation as a mechanism to regulate enzymatic activity, right? So in this case, if we phosphorylate, PFK2 becomes inactive. When PFK2 is not active, that means we are not doing glycolysis, we are doing gluconeogenesis. And based on what we discussed last class, when do we do gluconeogenesis? Starved state, because we don't have enough glucose, we need to find ways to make glucose. So we do gluconeogenesis. So in starved state, FBPS2 will be active. Which hormone regulates the activity of enzymes in the starved state? Glucagon. So this is under hormonal regulation by glucagon. Now, it's getting a little more complicated. So we talked about AMP activated kinase last class. But there are other mechanisms by which phosphorylation, dephosphorylation happens besides AMP activated kinase. And that is called cyclic AMP dependent protein kinase A pathway. Cyclic AMP, what is cyclic AMP? It's just like AMP, adenosine monophosphate, but we cyclize it. Cyclic AMP is produced as a signaling molecule, and these will activate the protein kinase A, which is, remember, kinase is a separate enzyme, right? So these will activate pKa. And this pKa is what will phosphorylate this enzyme. So that P is coming from pKa. So here, CAMP dependent pKa, and this is coming from pKa, protein kinase A. So glucagon is the hormone which is triggering this pathway to make protein kinase A active, and when protein kinase A is active, then it will phosphorylate this enzyme. So this P, you see that, is coming from pKa, protein kinase A. Remember, always kinases will add PI, right, using ATP. So this PI is added by protein kinase A to make this PFK2 inactive but FBPS2 active. So what about here? What enzyme do we need here? So this is under insulin. And which enzyme will this be? It's a phosphatase, and so it is actually called phosphoprotein phosphatase. So we have too many kinases, too many phosphatases we're dealing with right now. Total, we're looking at 3 kinases and 3 phosphatases. In the fed state, we have insulin, and insulin will trigger the hydrolysis of that phosphate so that we make PFK2 active, so that we can make F26BP, so that we can go on to do glycolysis by activating PFK1. In the starved state, we have glucagon, which will inactivate PFK2, activate FBPS2, so that we hydrolyze F26BP and do gluconeogenesis. So we have looked at, we only did PFK1, FBPS1. Now we still have pyruvate kinase. So that's the next thing we'll look at. You look at regulation of pyruvate kinase, which is one is going to be specifically for the liver. The other part is going to be for all other tissues. In liver, pyruvate kinase is under hormonal control. So what happened here, you see that in the dephosphorylated state, it is active. In the phosphorylated state, it is inactive. Similar to PFK2 here. PFK2 is inactive in the phosphorylated state, active in the dephosphorylated state. So now you have to tell me what hormone and how this will be regulated. So in the think about starved state. What does liver want to do? Make glucose. So do you want pyruvate kinase to be active or inactive in the fed state, in the starved state? Inactive, so that it doesn't make pyruvate, right? And use the glucose, export it to other tissues. So that's exactly what is happening. Pyruvate K is inactive in the liver in the phosphorylated state, and this is the same pKa, protein kinase A, and this is again under glucagon. It's under hormonal control in the liver by glucagon. It will activate the protein kinase A, which will phosphorylate it, making it inactive. And again, there's a phosphatase which will dephosphorylate pyruvate kinase, making it active. So same principle as we saw here. So you see the general theme, recurring theme, that phosphatases will promote what? Glycolytic pathways, right? And kinases are promoting gluconeogenetic pathways. Phosphatases are activated by insulin, kinases are activated by glucagon. And in all other tissues, what is happening? So here, of course, you have F16BP, and then we go to a lot of steps going to PEP, right? And from PEP we make pyruvate. So this is the irreversible step. Pyruvate kinase is needed to make that happen, PEP to pyruvate. So in all other tissues, pyruvate kinase is not under hormonal control. Only in the liver it is under hormonal control. All other tissues, it is under metabolite control. There are metabolites which controls its activity. So F16BP will allosterically upregulate the activity of pyruvate kinase, because if you have high F16BP, that means you have to make pyruvate. If you already have ATP, then why make pyruvate? So it will inhibit. What about acetyl-CoA? Where is acetyl-CoA made? Pyruvate oxidation. And acetyl-CoA is a signal. If you have high acetyl-CoA, that means that the cells have enough sources to get ATP, right? High acetyl-CoA, because from acetyl-CoA we get a lot of ATP, we will see. So all of these, both of these are going to actually deactivate or downregulate pyruvate kinase, both of these. So from pyruvate we can make alanine, only one step, we'll see how that happens. And so if you have high alanine, that means that it is also going to inhibit pyruvate kinase. The trigger here is that if you have high alanine, means that we must have high pyruvate. So if you have high pyruvate, don't make any more pyruvate. Again, in the liver, in the starved state, we want liver to make glucose available for the other tissues, right? So it does not want to do glycolysis for its own purpose. So that's why in the liver, in the starved state, glucagon will inactivate pyruvate kinase. We make acetyl-CoA. We haven't seen it yet, but we'll see. And how many carbons in pyruvate? 3. How many in acetyl-CoA? 2. So we must have lost 1 carbon by CO2. So this is highly hexagonic, right? Only one direction. No coming back. There's an enzyme which does this, and it's called pyruvate dehydrogenase complex. It is abbreviated as PDC. Okay, and what is the other thing that can happen from pyruvate? Yeah. Lactate under conditions when we have not enough oxygen, but let's assume we have enough oxygen. OA, right? And when we make OA from pyruvate, under what metabolic condition? To do what process? So this will for gluconeo. Acetyl-CoA will upregulate the activity of pyruvate carboxylase, and it will inhibit the activity of pyruvate dehydrogenase. So acetyl-CoA will, if you have high acetyl-CoA, then the trigger is don't make more acetyl-CoA, rather use pyruvate to make glucose to store it as glycogen or give it to other tissues. So acetyl-CoA will upregulate the activity of pyruvate carboxylase, so that go back, right, do the reverse process. And it will inhibit the pyruvate dehydrogenase complex, so that you don't make any more acetyl-CoA. So to simplify these things, you should always think about the what metabolic state and what enzymes are active under what metabolic state, fed state versus starved state. And so when you are fed, we are doing glycolysis, under starved, we are doing gluconeogenesis. And then these processes are reciprocally regulated to control those activities.